Preparation of standard working solution and sample. Centrifuge sample for 5 minutes at 1000 times gravity at 4 degrees. Centrifuge reference standard for 1 minute at 10,000 times gravity at 4 degrees. Add 1 ml of reference standard and sample diluent. Stand for 1 or 2 minutes. Add same volume like 500 microliter of reference standard and sample diluent to each tube. After standing, mix the standard thoroughly with a vortex meter of low speed. Centrifuge the solution for 1 minute at 800 times gravity. Take the same volume like 500 microliter of standard solution and dilute it. Mix thoroughly with a vortex meter. Dilute the standard in turn. The last tube is regarded as a blank. Preparation of biotinylated detection antibody working solution. Take 5,940 microliter of biotinylated antibody diluent. Add 60 microliter of concentrated biotinylated detection antibody to the diluent to prepare a 100-fold diluted working solution. Invert it gently for 20 times and set aside. Pipetting. Add 50 microliter of standard from low to high concentration. Avoid touching the inside wall. Vertically add in 50 microliter of prepared samples. Avoid touching the inside wall. Vertically add in 50 microliter of biotinylated detection antibody working solution. Avoid touch the inside wall. Cover the plate with sealer and mark the name, time, and abbreviation. Incubate at 37 degrees for 45 minutes. Preparation of one-time washing buffer. Take 288 ml of double distilled water. Add 12 ml of concentrated washing buffer to the double distilled water to prepare a 25-fold diluted working solution. Mix it with the magnetic stirrer for 5 to 10 minutes. Preparation of the HRP Conjugate working solution. Take 11,880 microliter of HRP Enzyme Conjugate diluent. Add 120 microliter of concentrated to the diluent to prepare a 100-fold diluted working solution. Invert it gently for 20 times and set aside. Take out the plate. Avoid touching the bottom of the well. Avoid touching the bottom of the wells. Washing. Pat it dry and replace the absorbent paper. Vertically add 100 microliter per well HRP Conjugate working solution. Cover the plate with sealer and mark the name, time, and abbreviation. Incubate at 37 degrees for 30 minutes. Take out the plate. Avoid touching the bottom of the wells. Washing. Pat it dry and replace the absorbent paper. Color development. Vertically add 90 microliter per well substrate reagent. Cover the plates with sealer and mark the name, time, and abbreviation. Incubate at 37 degrees for about 15 minutes. Take out the plate. Avoid touching the bottom of the wells. The last 4 wells will show a clear blue gradient after color development. Reaction terminated. Vertically add 50 microliter per well stop solution. The color turns yellow from blue when the stop solution is added. Data statistic. Gently wipe the bottom of the plate. Put the plate into the microplate reader and determine the optical density.